So to address both of these problems, we'll be now diving into the technologies and how by using these technologies we can generate data, and that data can help us answer those questions. And so the first technology that we'll be talking about is a technology called single-cell RNA sequencing. This emerged in the last decade or so and has really revolutionized our ability to understand cell biology and understand disease. The second problem is that we know how a few of these genes worked. Like, for example, we learned that PDGFRA is important for cell division, so if you delete this gene, then the pre-adipocytes won't proliferate anymore. And AdipoQ is very important for the adipocyte population, and so maybe if we delete this gene, then we'll see less productive signaling between adipose tissue and other tissues. But the question is, can we learn for all the remaining genes out of the 20,000 or so genes in our genome what they too are doing? And if we had a technology that would allow us to delete these other genes and then see what the impact is on the cells, then that would really help us towards this goal. And so in parts B and C, we'll be talking about technologies that really address this first challenge of measuring every gene in every cell to understand which genes are switched on or off in which cells. And secondly, perturbing these genes or deleting these genes to see what happens to the cells when those genes are deleted. So talking about the first technology, which we use to measure gene expression in cells, this is a technology called single-cell RNA sequencing. It's really revolutionized our ability to do cell biology and to understand the cellular states that drive disease. And so to simply put, what happens with single-cell RNA sequencing is that you start with a population of cells of interest, and from these you can measure in every single cell the activity level of every single gene. And so the output that you get is this kind of matrix where every row is a different gene and every column is a different cell. So you would have maybe 20,000 genes for every gene in the genome, and however many cells you profile in that experiment. So for one of the data sets in the first crunch, this is roughly 150,000. And then each value tells us how many RNA molecules, or in other words, how active every gene is in each of these different cells. And so going back to some of the cell populations and programs that we're interested in, if this is the pool of cells that we perform single-cell RNA-seq on, what we might see when we look at the resulting matrix is that the genes PDGFRA and Thy1 are switched on in these two first cells, and so probably then these two first cells are pre-adipocytes. And we might also see that the genes AdipoQ and FABP4 are switched on in these 4 subsequent cells, and so we might conclude from that that these cells 3 to 6 are classic adipocytes. And finally, we might see that these 2 lipogenic markers, SREBP1 and FASN, are specifically switched on in a subset of the adipocytes in the last 2 cells, 5 and 6. And so we might say that these are particularly lipogenic cells among the larger adipocyte population. And so, as I mentioned, we can profile hundreds of thousands of cells with this technology. And actually, this is very exciting because the last 15 years or so, since the very first single-cell RNA-seq technology was published, has seen enormous growth. And so we went from a case where in 2009, 4 cells in a 4-stage blastomere were sequenced. So in the matrix on the previous page, there were just 4 columns. to today, this is the logarithmic scale and showing the number of cells, to the case where today, or at least 8 years ago, in 2017, we now have technologies that allow us to profile 1 million cells. And so now I'll switch gears a little bit and tell you about how this single-cell RNA-seq experiment actually works. This isn't information that you'll directly need to do the challenges, but it's very helpful, I think, for you to understand. And so what we start from is a population of cells. So this can either be cells that are found in a piece of tissue or cells that we've already grown in a dish in the lab. And cells tend to interact and to communicate. And so the first step is to actually go from this population to a dissociated version of this population where every cell is no longer kind of attached to other cells. We then take these cells and isolate them one by one. And for each of them, we can then count how many of each gene's corresponding RNA molecules are found in that particular cell. So it's this workflow again where we start from a kind of population of cells, dissociate them, and then one by one are able to measure the RNA molecules inside each of those, therefore telling us about the genes which are active there and the gene expression programs in particular which are expressed. And so I'll tell you a little bit more now about this isolation step and how that works. So basically what happens is we have this microfluidic system and cells are flown in through here into this chamber. And then they become encapsulated in individual water droplets. And what you can see is that in each of these water droplets, along with the cell, you also have a colored bead that's encapsulated with it. And this colored bead will act as a barcode so that we'll be able to tell for all of the RNA molecules inside every cell which barcode it had. So for example, this cell 3 here has these 4 RNA molecules which are shown in red because this cell was encapsulated with the red bead. And so that bead allowed us to barcode these individual RNA molecules. Okay, so that's this method for isolating cells. And the reason that we want to isolate the cells is so that we can individually barcode all of the RNA transcripts that come from a particular cell with that barcode. And now we can go back to our overall workflow where we've done the dissociation, the isolation, the labeling of the RNA molecules. And what actually happens now is the reverse of what typically happens in the central dogma in biology, which is where DNA is transcribed and produces RNA. So here we actually do the reverse process, a process called reverse transcription, where we go from the RNA, and for each RNA molecule, we convert it to the corresponding DNA molecule. And we do this for 2 reasons. Firstly, mainly we do this because DNA is very stable and RNA is not. And secondly, this then allows us to actually amplify these molecules of DNA. And so we have a much larger pool of DNA molecules that kind of accurately represent the proportions of RNA molecules that we originally detected. And once we have these DNA molecules, we can then do sequencing, which is basically saying for every single one of these DNA sequences, going through and seeing which of the 4 letters in the genetic code, A, C, T, and G, are at that position in each DNA sequence. And so why is that helpful? What do we then do with this kind of sequencing library? Well, we now have the Human Genome Project. This was completed in 2003. And this was a really amazing effort that produced the sequence of every gene in the human body. And so now we have these sequences of DNA that correspond to scrap that bit. And so now we have these sequences of DNA. What we can do is we can actually go back to the human genome and find out which gene each of these sequences comes from. And so once we've done that, we can now count within this sequencing library how many sequences we see corresponding to each of these genes in the human genome. And yeah, so this is the expression matrix or the output of the single-cell RNA-seq experiment that we've seen already, and is kind of the main data structure that you'll be working with through these crunches. So just a couple more things to be aware of with these matrices. So first of all, just to reiterate, every row is a different gene and every column is a different cell. And then the values represent the activity level of that gene in that cell. And so, but one issue with these matrices, with the output of the single-cell RNA-seq experiments, is that they tend to be quite sparse, meaning that there are many entries here where the value is actually 0. And this is a bit complicated because in those cases, we can't actually distinguish between two interpretations, one being biological, that in reality, this gene is actually 0 in the cell, and one being just technical, that that gene was not detected at high enough abundance to be captured in the experiment. And so basically, the kind of pre-processing in this bioinformatics pipeline involves dealing with this issue. And so this is typically how we deal with that. We want to remove cells that have too many zeros and therefore seem low quality. And so what you see here on the x-axis is the cell basically ordered by this ranking of how many genes have a value of 0 in that cell. This is what we typically call the number of detected genes. And on the y-axis, so on the x-axis is this rank of the cells, and on the y-axis is the number of genes that were detected, so the number of non-zero genes. And so what we tend to do is to remove this lower tail of cells that have too few genes detected, and so are probably not very helpful for downstream analysis. And this is another issue now, but we actually also tend to remove the cells which are at the top end of this, because often these have a suspiciously high number of genes detected in the cell. And what that suggests is that the isolation step that we spoke about previously is actually not fully working. And maybe rather than an individual cell being in one of those water droplets, we actually have 2 or multiple cells in there. So we remove these two ends, and we're left with these high-quality cells in the middle here for our analysis. Okay, so this is how we deal with the low-quality cells in the data. And then there is also a second issue that we have to deal with, which is that the total number of RNAs that we count in individual cells might be different from cell to cell. So in other words, some cells might have higher overall gene expression, but this is actually just because of technical reasons. And so we'd like to normalize for that. And so what we do is we scale the gene expression per cell such that it sums to 100,000, which is roughly how many molecules of RNA we might expect a given cell to have. And then we add a pseudo count of 1, and then log transform the data so that our distribution is Gaussian, and this makes things much easier for downstream analysis. And then finally, in this third step, we now have normalized cells, only high-quality cells. We can project these cells onto a lower dimensionality embedding, and so this is one way using, for example, UMAP or TSNE, that we typically visualize the single-cell RNA-seq data set and the kind of global diversity of cells that we see there. And so going back to our particular use case, this is one of the data sets that you'll be working with, an example of this lower dimensional embedding of the cells. And you can see here that we've marked which of the cells are pre-adipocytes and which of the cells are adipocytes. And again, we've done this because now that we have the single cell RNA-seq data, we can actually go and see which genes are expressed in which cells. And what we find is that the classical gene markers of the pre-adipocytes, as well as other genes in the pre-adipocyte program, are expressed in these cells, while the classical markers of the adipocyte population, AdipoQ and FABP4, as well as other adipocyte markers, are expressed in this pink population.